Macromolecules contain a specific sequence of subunits. Although the chemical reactions for adding subunits to each polymer are different in detail for proteins, nucleic acids, and polysaccharides, they share important features. Each polymer grows by the addition of a monomer onto one end of the polymer chain via a condensation reaction, in which a molecule of water is lost with each subunit added. Figure 228. See also Figure 217. In all cases, the reactions are catalyzed by specific enzymes, which ensure that only monomers of the appropriate type are incorporated. The stepwise polymerization of monomers into a long chain is a simple way to manufacture a large, complex molecule, because the subunits are added by the same reaction performed over and over again by the same set of enzymes. In a sense, the process resembles the repetitive operation of a machine in a factory, with some important differences. First, apart from some of the polysaccharides, most macromolecules are made from a set of monomers that are slightly different from one another. For example, the 20 different amino acids from which proteins are made. See panel 2.5, pages 72 to 73. Second, and most important, the polymer chain is not assembled at random from these subunits. Instead, the subunits are added in a particular order, or sequence. The mechanisms that specify polymer sequence in a cell are discussed in chapters 6 and 7. These mechanisms are central to biology because the biological functions of proteins, nucleic acids, and many polysaccharides are absolutely dependent on a particular sequence of subunits in the linear chains. The possibility of varying the sequence of subunits creates enormous diversity in the polymeric molecules that can be produced. Thus, for a protein chain 200 amino acids long, there are 20 to the power of 20 possible combinations. 20 times 20 times 20 times 20 multiplied 200 times. While for a DNA molecule, 10,000 nucleotides long, small by DNA standards, with its 4 different nucleotides, there are 4 to the power of 10,000 different possibilities, an unimaginably large number. Thus, the machinery of polymerization must be subject to a sensitive control that allows it to specify exactly which subunit should be added next to the growing polymer end. Non-covalent bonds specify the precise shape of a macromolecule. Most of the single covalent bonds in a macromolecule allow rotation of the atoms they join, so that the polymer chain has great flexibility. In principle, this allows a macromolecule to adopt an almost unlimited number of shapes or conformations, as the polymer chain writhes and rotates under the influence of random thermal energy. However, the shapes of most biological macromolecules are highly constrained because of weaker non-covalent bonds that form between the different parts of the molecule. If these weaker bonds are formed in sufficient numbers, the polymer chain can preferentially adopt one particular conformation, determined by the linear sequence of monomers in its chain. Most protein molecules and many of the RNA molecules found in cells fold tightly into one highly preferred conformation in this way. Figure 231. These unique conformations, shaped by natural selection, determine the chemistry and activity of these macromolecules and dictate their interactions with other biological molecules. The non-covalent bonds important in biological molecules include 2 types described earlier in this chapter: electrostatic attractions and hydrogen bonds. See panel 2.7, pages 76 to 77. Electrostatic attractions, although strong on their own, are quite weak in water. This is because charged or partially charged polar groups are shielded by their interactions with water molecules or with other salts present in the aqueous solution. Electrostatic attractions, however, are very important in biological systems. An enzyme that binds a positively charged substrate will often use a negatively charged amino acid side chain to guide its substrate into the proper position. And we have already mentioned the importance of hydrogen bonds in establishing the unique properties of water. Hydrogen bonds also hold the two strands of the DNA double helix together. Because individual hydrogen bonds are weak, enzymes can easily unzip the helix, for example, when a cell needs to copy its genetic material. A third type of weak bond results from van der Waals attractions, which are a form of electrical attraction caused by fluctuating electric charges that arise whenever two atoms come within a very short distance of each other. Although van der Waals attractions are weaker than hydrogen bonds, in large numbers they play an important role in the attraction between large molecules with complementary shapes. All of these non-covalent forces are reviewed in panel 2.7. Another important non-covalent force is created by the three-dimensional structure of water, which forces hydrophobic groups together in order to minimize their disruptive effect on the hydrogen-bonded network of water molecules. See panel 2.7 and panel 2.2, pages 66 to 67. This expulsion from the aqueous solution generates what is sometimes thought of as a fourth kind of weak non-covalent bond, called a hydrophobic interaction. This interaction forces phospholipid molecules together in cell membranes, and it also gives most protein molecules a compact globular shape. Non-covalent bonds allow a macromolecule to bind other selected molecules. Although non-covalent bonds are individually very weak, they can add up to create a strong attraction between two molecules when these molecules fit together very closely, like a hand in a glove, with many non-covalent bonds between them. See panel 2.7, pages 76 to 77. This form of molecular interaction provides for great specificity in the binding of macromolecules to other small and large molecules, because the multi-point contacts required for strong binding make it possible for a macromolecule to select, through binding interactions, just one of the many thousands of different molecules present inside a cell. Moreover, because the strength of the binding depends on the number of non-covalent bonds that are formed, interactions of almost any strength are possible. Binding of this type underlies all biological catalysis, making it possible for proteins to function as enzymes. Non-covalent bonds can also stabilize associations between two different macromolecules if their surfaces match closely. Figure 232 and movie 2.4. These bonds thereby allow macromolecules to be used as building blocks for the formation of much larger structures. For example, proteins often bind together into multi-protein complexes, thereby forming intricate machines with multiple moving parts that perform such complex tasks as DNA replication and protein synthesis. Figure 233. Thus, non-covalent bonds account for much of the specificity that we associate with living cells. Essential concepts. Living cells obey the same chemical and physical laws as non-living things. Like all other forms of matter, they are composed of atoms, which are the smallest units of a chemical element that retains the distinctive chemical properties of that element. Atoms are made up of smaller particles. The nucleus of an atom contains protons, which are positively charged, and uncharged neutrons. The nucleus is surrounded by a cloud of negatively charged electrons. The number of electrons in an atom is equal to the number of protons in its nucleus. The nuclei of different isotopes of the same element contain the same number of protons but different numbers of neutrons. Living cells are made up of a limited number of elements, 4 of which, C, H, and O, make up 96.5% of their mass. The chemical properties of an atom are determined by the number and arrangement of its electrons. An atom is most stable when all of its electrons are at their lowest possible energy level and when each electron shell is completely filled. Chemical bonds form between atoms as electrons move to reach a more stable arrangement. Clusters of 2 or more atoms held together by chemical bonds are known as molecules. When an electron jumps from one atom to another, 2 ions of opposite charge are generated. Ionic bonds can then arise by the mutual attraction of these charged atoms. A covalent bond consists of a pair of electrons shared between 2 adjacent atoms. If 2 pairs of electrons are shared, a double bond is formed. Living organisms contain a distinctive and restricted set of small carbon-based molecules that are essentially the same for every living species. The main categories are sugars, fatty acids, amino acids, and nucleotides. Sugars are a primary source of chemical energy for cells and can be incorporated into polysaccharides for energy storage. Fatty acids are also important for energy storage, but their most essential function is in the formation of cell membranes. The vast majority of the dry mass of a cell consists of macromolecules, formed as polymers of sugars, amino acids, or nucleotides. Macromolecules are intermediate in both size and complexity between small molecules and cell organelles. They have many remarkable properties that are not easily deduced from the subunits from which they are made. The remarkably diverse and versatile class of macromolecules known as proteins are polymers formed from amino acids. Nucleotides play a central part in energy transfer and are the subunits from which the informational macromolecules RNA and DNA are made. Protein, RNA, and DNA molecules are synthesized from subunits by repetitive condensation reactions. Each of these biological macromolecules has a unique sequence of subunits. Weak non-covalent bonds form between different regions of a macromolecule. These can cause the macromolecule to fold into a unique three-dimensional shape, conformation, with a special chemistry, as seen most conspicuously in proteins.